pleased to announce our first speaker, Arlan Rodrigues. He's a postdoc in the Department of Animal Dairy Sciences at UW-Madison, working in Hilário Mantovani's lab. Please welcome Arlan to the stage.
So, hello everybody, my name is Arlan, and today I'm going to present our research on the enterosignature dynamic of healthy and diarrheic dairy calves during the pre-weaning period. So in this study, I'm going to show how the changes in the gut microbiota may help us to understand the development of diarrhea in early life calves. So let's get started. Neonatal calf diarrhea remain a big challenge for the dairy industry. It's a leading cause of economic losses in the dairy system, and we estimate that about 50% of the sick animals is sick due to diarrhea, and easily 20% of the early life deaths is caused by diarrhea. Beyond that, early life diarrhea has also one long-term impact in the growth and in the overall productivity. Easily, we can distinguish a happy calf. It's active and playful animal from a sad calf. It's always is quiet, tired, and weak, and it's always struggling with diarrhea and dehydration. So, understanding the factors associated with the calf health, especially during the first week of life, is very, very important. And one of the factors that I would like to talk today with you is the early life gut microbiota. So, because it's play a critical role in these outcomes. The fast microbial colonization after birth is essential for building the immune system, for manage the nutrient and metabolism, and for maintain the gut body function. However, when this process is disrupted, I said also call it as a dysbiosis, the animals become much more susceptible to disease. So understand how these bugs evolves is a good opportunity for early life interventions. And nowadays, much of our knowledge is based on the snapshot profile. We usually collect samples at a single time point, which provides us one static view of the gut microbiota. And this is good and very useful. However, we miss one important detail, the longitudinal dynamic of the gut microbiota. And what we need to do is think as a longitudinal perspective. We have to, we have to follow a set of animals or a group of animals over time and collect samples to capture the temporal dynamic of the gut microbiota and measure the microbiome maturation patterns. And this brings us to the enterosignature concept. The enterosignature is the biological signature of the gut. The idea here is identify common bacterial guts, which may have a potential to be beneficial or harmful to the animal. And we can calculate that using a machine learning approach, applying the non-negative matrix factorization. We can start using the genus level or species levels, depending how deep is your sequencing, using one abundance relative matrix table. And you have to estimate two matrices, the W matrix, which is the enterosignature, those microbes that occur together across the sample or they appear together across the samples, and finally estimate the relative abundance of each enterosignature in the samples. So, and the beauty behind this analysis is that instead we handle one massive data with hundreds and hundreds of individual bacteria, you can narrow it down to a few meaningful enterosignature, and we can be able to analyze our data. So in this context, the objective of this study is identifying the fecal enterosignature during the pre-weaning period and evaluate how they change over time in healthy versus diarrheic dairy calves. So to do that, we monitor 56 animals over 8 weeks, and we collect a total of 244 fecal swab samples. So we collect the samples at week 1, 2, 4, 6, and 8, and we divided the animals into experimental groups. And healthy group, no diarrhea at all, and a diarrheic group, one or more episodes of diarrhea. So at the end, we had 26 animals in the healthy group and 30 animals in the diarrheic group. We also measured the feed intake and body weight of those animals, and as well as the fecal score based on this scale: 0 as the solid feces and 3 as a wet feces. After that, we did the DNA extraction and sequenced the samples. Following, we analyzed the data and applied the enterosignature method. For the statistical analysis, we are comparing the differences between the relative abundance of the enterosignature across the weeks or over time, and the differences between groups, healthy versus diarrheic calves. So the first result is about feed intake and body weight. So the green line represents in the right, we have the body weight, and in the left, we have the feed intake. And the green line represents the healthy group, and red represents the diarrheic group. So in summary, the healthy group had a high body weight and feed intake in comparison to the diarrheic group. And we also noticed that after the second week, the body weight diverged, and the feed intake diverged after the fourth week. So these results support the idea that diarrhea is not one point problem. It has a longitudinal impact on the animals. But the point is, how does the gut microbiota can impact or influence these results? And we can see that analyze the enterosignature. We identified 5 enterosignatures, which explain 87% of the microbiome variation. So the figure shows the composition of each enterosignature of the top 5 genera. And as we can see, each enterosignature has its own microbial profile. And the task here is identify those enterosignatures with a potential to be beneficial or harmful to the animal. So let's break it down and understand it better. So the enterosignature number 1 is dominated by Prevotella, which is a genus associated with carbohydrate fermentation and volatile fatty acid production, and with a small contribution with Succinea vibrio. It's a VFA producer as well. We also can see here one small contribution of the Fecalium bacterium, which is linked with anti-inflammatory response. So analyzing this enterosignature, we can target it as a potential beneficial enterosignature to the animal. Following to the ES2, we can see there is a dominated by the Ficaicola genus, which is a core commensal genus, but we also have one small contribution of the Fecalium bacteria linked to anti-inflammatory response. So here we can target as a neutral enterosignature. Moving ahead for the ES3, we can see a dominance by bacteroids, But you also can see one contribution of a lactobacillus, which is one a beneficial genus, and the Fecalium bacterium linked to inflammatory response. This enterosignature in particular has also a good potential to be a beneficial enterosignature to the animals. The ES4, however, has quite interesting composition, because we can see here Clostridium, and nobody wants to see Clostridium in the animals because it's correlated with inflammatory response and produce some bad toxins. We can also see Enterococcus and Streptococcus, which can be bad for the animals as well. And the ES5 is codomined by Fusiobacterium and Prevotella massiva. Fusiobacterium is associated with inflammatory response, whereas Prevotella massiva is a less characterized genus. So once we identified the enterosignature, let's move to understand the relative abundance and the longitudinal dynamic. Here is the relative abundance of the whole set of samples, 244 samples, and here we have the average of this figure. So I have 2 points to mention that. The first one, we got a huge variation in the enterosignature relative abundance in the samples, but even so, we could see some patterns. We notice here one increase in some enterosignature as ES1 and ES5. ES2 kept consistent over time, while the ES3 decreased. ES3 and 4 decrease over time. So let's break it down and understand those numbers. So the ES1 associated with a carbohydrate fermentation and volatility producer increase over time. And this makes sense because as the animal grows, the hormone becomes functional, and they need such microorganisms to colonize the hormone. And the ES2 didn't change over time, where the ES3 decreased. Moving ahead with the ES4, we could see one decrease in both groups, and the ES5 increase. But yeah, in overall, the dynamic of the enterosignature over time is quite similar in healthy and diarrheic group. However, our most significant result was at week 1, when the enterosignature was quite different. We can see here that the enterosignature 4, specifically the 4 composed by those genes with a potential to be harmful to the animal, was much higher in the diarrheic group in comparison to the healthy group. And the enterosignature 5 was much higher in the healthy group in comparison to the diarrheic group. So to conclude, the neonatal diarrhea is associated with different enterosignatures, and the largest difference was observed at week 1, the first week of life, and after that, the ES tends to be stable, and the first week of life is the key, is a critical point for target intervention. So I would like to say thank you for the fund sources to make this project happen, as well as for my lab mates in the Mantovani labs. So thank you so much for the audience, and I'm ready to take any questions.
Questions?
Yes, the floor is open for questions. We have one over here.
Good afternoon. I really appreciated your presentation. For your calves that had diarrhea, are you noticing, or do you have any data on specific pathogens that may be associated with different fingerprints, like whether in our first week of life, Enterotoxigenic E. coli or Salmonella and then Crypto coming in?
So we did only the syncytials, but we don't have. I'm not sure if you have those other pathogens like Cryptosporidium or Emeriosis. So, but for the bacteria, yes, we do.
Other questions? Over here.
Thank you, very interesting. How do you think the type of diet that the calves are eating would affect these enterosignatures?
Oh, thank you for the question. That's a good question. The diet for sure influence, and we could see this in the dynamic of especially the ES1, when we have Prevotella is associated with carbohydrate and volatile fatty acid production. And yeah, if you change the diet, maybe, I don't know, applying a cocktail with a specific microorganism, a good or a prebiotic or a pre or probiotic, maybe just change the dynamic of the enterosignatures.
Other questions? We have time.
I'll ask one. May I?
Yes, absolutely. Amanda, go ahead.
Thank you for the presentation. I was wondering if you have any data showing that recurrent cases might change the signature. So you mentioned that these calves were at least one case over the 8 weeks. Do you know if it will change if a calf has, 2 events or 3 over the course of?
Yes, and at the beginning we divided the animals in 3 groups. Was the healthy, no diarrhea, resilient, 1 case of diarrhea, and a susceptible, 2 or more cases of diarrhea. And we noticed that the resilient and hesitant tends to have the same pattern. So that's why we decided to combine both groups and working only with healthy and a diarrheic group.
Thank you.
All right. Any other questions? I think I have a question as well. So I'm a reproductive biologist. We have postpartum dairy cows that are all in the same environment, post-calving, and some get metritis and some don't. But they're all exposed to the same environment. I mean, is it the same situation with your calves? I mean, they're all in the same environment, they're all getting the same diet, yet some get diarrhea and some don't. Is that correct?
Yes, it's correct, but as you could see, the environment is important, I agree, but I noticed with this research is that the gut play one fundamental role, and especially in the first week, when we have the high composition of Clostridium, Enterococcus, or Streptococcus, those animals tend to have one more incidence of diarrhea later on, and this impact the growth and the feed intake and body weight, for example, as I showed.
Okay. Last chance for a question. If not, let's give Arlan a big round of applause.